So this week and next we'll talk about transgenics and biotechnology, right? And one concept that I wanted to introduce real quick is the difference between what we know as forward genetics versus reverse genetics. So forward or traditional genetics is what people like Mendel did. They would be interested in a given phenotype and then they would try and look for what mutations were behind those phenotypes of interest. In time, if someone was interested in the genes that controlled a given trait or characteristic, they would focus on that characteristic, induce mutations by subjecting individuals, plants or animals to X-rays or chemical mutagens, and then look for those individuals that show changes to their phenotype of interest, and then ask the question, what genes are behind this? So forward or classical genetics was we start with the phenotype and then we ask questions about what genes are behind this phenotype. But then came our ability to reverse this process and then start with a gene that we know and ask the question, what does this gene do? What is the phenotype that will that overexpressing this gene or mutating this gene will cause? So it was reversing that process. Instead of starting from the phenotype and asking questions about the genotype underlying that phenotype, it was starting from the gene and then asking questions about what changing it would do to the individual. So we do normally that through transfections in cell culture, that is introducing different versions or mutants or genetic constructs related to a given gene of interest. We call them transfections if we're dealing with eukaryotic cells. We call them transformations if we're dealing with bacteria, with prokaryotes. And then we can take it a step further and basically genetically modified an entire organism, in which case we're not talking about transfections or transformations anymore, we're talking about transgenic organisms. And if we happen to take out a gene from an organism, we refer to those as knockouts, and we'll describe both today. Let me ask you a question. Let's say that this is a vector that you will introduce in bacteria. We have our multiple cloning site that we discussed last week. We have our selection marker, in this case ampicillin resistance, and it turns out that our MCS or multiple cloning site is embedded in the LAGZ gene that would allow us to do that blue-white transformation that we discussed last week. Now, let's say that we would like to express high levels of whatever DNA we introduce into this vector once this vector is inside of bacteria. What do you think this vector would need if you wanted to express a lot of the gene that you're introducing into this bacteria? That's right, a promoter, right? So that way the cell knows that this is a gene that needs to be transcribed. And what is a promoter like in bacteria? Do you remember? The Shine-Dalgarno is part of it, if you will, something close. It's that famous minus 35 and minus 10 sequences, right? So if you want this to be a bacterial expression vector, upstream of the MCS, you need a very strong minus 35, minus 10 consensus sequence, and also a strong SD, which stands for Shine-Dalgarno. And if you guys remember, the Shine-Dalgarno is a sequence that allows ribosomes to bind bacterial mRNAs and initiate translation, or rather the scanning for the AUG and eventually translation. So the idea here is that if the sequence of DNA that you're introducing into bacteria you would like to express at very high levels, you need to tell the bacterium, this is a gene that I like to express. So you need that promoter and a Shine-Dalgarno sequence, okay? What would a eukaryotic expression vector look like then? What would you need to put in a vector if you wanted to express your insert of interest in eukaryotes? Same thing, right? You need some sort of eukaryotic promoter to tell the cells, whichever cells you're introducing this into by transfection, that you would like to express this gene, right? So same thing, what you would need is a strong eukaryotic promoter, and often what we do is we use viral promoters. And there is a reason for that. There is a biological reason for that. When viruses infect eukaryotic cells, let's say our cells, they immediately go to work, right? All they want to do is replicate, make many copies of themselves, and then eventually burst out of that infected cell and infect others. So viral promoters tend to be exceedingly potent. So if you introduce a viral promoter just upstream of the sequence that you're cloning into these eukaryotic expression vectors, chances are that when this vector, with your sequence of interest, makes it into a eukaryotic cell, those cells will immediately start expressing very high levels of it. And the one thing that is not shown here, I'm sorry, I thought that I had that slide, but you also need just downstream of your cloning site a poly-A signal. Because if the DNA that you're introducing into this vector you would like to express in eukaryotic cells, those eukaryotic cells need to add a poly-A tail to whatever RNA is being transcribed for that RNA to be translated. If it doesn't have a poly-A tail, it won't be processed the way the mRNAs are to be properly translated, okay? You don't have to worry about the 5' cap because that's something that RNA polymerase and the initiation complex take care for themselves, but you do need to provide a very strong poly-A signal so that when your insert of interest is transcribed into RNA, a poly-A tail is added and that DNA can be translated. Any questions so far? So last week we left it at how we introduce DNAs of interest into these vectors and how we introduce these vectors into cells. Now we're taking a step further, and what we're basically introducing is this idea that depending on what you'd like to do with those sequence of interest inside of cells or organisms, you may need to add a few more things to these vectors so that they can either express a DNA into an RNA that may be eventually translated, or whatever the objective may be, you may need to introduce that in the vector before you place your DNA of interest into them. Oh, now I see what happened. I had it as one of those slides that would reveal themselves so that I don't give it away. Okay, so it'll show up in the annotated slides later, but you'll see a cartoon of what a eukaryotic expression vector looks like with a strong poly-A signal and a strong eukaryotic promoter. These eukaryotic expression vectors still have a bacterial selection marker, so let's say an ampicillin resistance gene, and a bacterial ORI sequence. And one of the questions that I was gonna ask if I could reveal this slide is, why do you think we have those? I'm gonna ask the question and answer it myself. So the reason why we keep this stuff that is useful in bacteria is because even though the final destination of these eukaryotic expression vectors is eukaryotic cells, we normally pass them through bacteria first. Because all of the early steps in cloning and making sure that your insert of interest made it in one piece and in the right orientation, and that you can make many copies of these plasmids so that you can later introduce it into eukaryotic cells, all of that is much easier done in bacteria. So it's no, even though your final destination may be a eukaryotic cell, all of those early steps we do in bacteria. It's much easier. Faster, cleaner, cheaper, everything, right? So that is why you'll see that in eukaryotic expression vectors, there is still some bacterial stuff like ampicillin resistance and bacterial ORI sequences. So let's say that you cloned your sequence of interest into one of these eukaryotic expression vectors, and like I just said, you would take the first early steps in bacteria. So you go through the process of transforming bacteria, plating your bacteria on a plate with ampicillin, and then hope that some colonies grow. And you would know that those colonies represent bacteria that took up a plasmid carrying the ampicillin resistance gene. Right? What we don't know, what you don't know, as we discussed last week, is what kind of vector made it into those cells, into those bacteria. We're still in bacteria. It could be empty vector that recircularized or was never really fully digested by the enzyme that you use for cloning. It could be the vector with insert in the right orientation, or it could be the vector with insert but in the reverse orientation. And here's where this business of right and versus reverse orientation makes a little more sense. If you have a strong eukaryotic promoter and you end up with your insert of interest in the reverse orientation, that promoter will direct the transcription of a gene that is backwards. And whatever you're going to get out of that is most likely nonsense, right? So that's why sometimes you need your insert of interest to go in a given orientation, namely the orientation for translation of that sequence to give you the protein that you want. So we want a vector that has the insert in the right orientation, and as we discussed last week, there's a number of approaches that you can take. One of them is going for a diagnostic restriction enzyme digest, by which you strategically choose an enzyme that might have target sites outside and inside of your insert. And depending on the binding pattern that you expect, depending on whether your insert went in the right or the wrong orientation, you can run the digest and then choose whatever clones, that's what we would call one of those colonies that we pick up from the plate, whatever clones have the insert in the right orientation. So here in this example, we have the first lane would correspond to an empty vector that made it into the bacteria. The second lane is the one we want because it has the vector and an insert that seems to have gone in the right orientation, but then the last lane would represent one with the vector and the insert in the wrong orientation. Or we could do diagnostic PCR. We spent some time last week discussing how, depending on how you choose your primers, you can make it such that the only time that you would get a PCR amplification product is if your insert was cloned into the vector and in the right orientation. No clone or insert in the wrong orientation, and you don't get an amplicon. So in this case, once again, the second clone would show a strong PCR band. Neither of the other two reactions would show amplification. So all of this we would do still in bacteria, and that's why even eukaryotic expression vectors still have things like ampicillin resistance and bacterial ORI sequences, because you need to work with them in bacteria for a while. That actually makes room for a concept that is kind of special. It's the concept of shuttle vectors, and that is when we have a bacterial vector that may have your strong bacterial promoter and Shandak-Garno sequence and your LAG-Z for blue-white selection. That's fine, but you have your bacterial vector, and you may use that bacterial vector for insertion using one restriction enzyme, right? So in bacteria, you would go through the whole trick of finding out if the insert made it in or not, and if it did, whether it's in right orientation or not. But then what you do is you take that vector with the insert in the right orientation, and you use now two restriction enzymes to move it to your eukaryotic expression vector, knowing that because you're using two restriction enzymes, there's going to be directional cloning and you can't get it wrong. So the reason we sometimes use these shuttle vectors, which are simply bacterial vectors that we don't ever use to express our sequence of interest in bacteria, we just use them out of convenience, because it's so much easier to determine if everything is right in bacteria. And then with a simple restriction digest with two enzymes, you can move it into a eukaryotic expression vector directionally. And one of the reasons we do that is sometimes these eukaryotic expression vectors have multiple cloning sites that are a little more limited than those in bacteria. In bacteria, most vectors for bacterial transformation have plenty of restriction enzyme sites, whereas Eukaryotic ones may be a little more restricted. So you go through a bacterial one out of convenience, you figure that everything is okay, and now you can choose any combination of two enzymes that allows you to carry out directional cloning into your expression vector of interest. I just wanted to mention that because you can come across this idea of shadow vectors, and that's all it means. It's an intermediate vector that you just use out of convenience. All right, so now let's say that you have your eukaryotic expression vector with your insert of interest in the right orientation, and you put it in cells. And what I'd like to do now is discuss this concept of transient versus stable transfections. And before I do that, I need to briefly explain the following. In bacteria, when you put a plasmid into bacteria, plasmids that have a strong ORI sequence can replicate themselves during the life cycle of bacteria, such that anytime a bacterium divides, both daughter cells inherit a copy of the plasmid. For all intents and purposes, those plasmids are, for bacteria, like their chromosomal DNA. They're going to be there forever, right? Because with every cell division, there's going to be a replication of the plasmid, and both daughter cells will inherit a copy. In some cases, you can have high copy number plasmids, in which case the daughter cells inherit more than one copy of these plasmids. But eukaryotic cells are not like that. Eukaryotic cells don't have plasmids hanging around naturally, right? If a eukaryotic cell sees a small piece of circular DNA, immediately it thinks it's some sort of foreign agent trying to infect it. So chances are that it may attack it, right? So plasmids can't survive for too long in eukaryotic cells. So you can use them to introduce your DNA of interest into the cells, but once those plasmids have made it into the cells, they're not going to hang out for long. In fact, anytime that eukaryotic cell divides, the plasmid won't, so immediately you will have this process of plasmid dilution that we discussed last time. Because even though the vector that you're using may have a strong bacterial ORI sequence, eukaryotic cells can't do anything with it. Okay? Now, even a short survival of the plasmid inside of cells may still be useful to you. Mostly in research contexts, what happens often is we introduce the plasmids into the cells. Not all cells will take up the plasmid because there's always a component of luck in having these plasmids go into cells. It's ever so slightly easier, particularly with animal cells, because they don't have a cell wall like bacteria do. So it's sometimes a bit easier for the plasmids to go into animal cells in culture, but not all of them will take it up. And we know that after 24, 48, 72 hours, the cells will divide, the plasmids won't, so there's going to be plasmid dilution. But one thing we can do is something called transient transfection, and that is we let the plasmid express whatever it carries, and then if we have something elsewhere in the same plasmid that allows us to identify those cells that picked up the plasmid, then we can simply try and identify those cells based on that extra marker, and then assume that those cells picked up the plasmid and therefore are expressing our DNA of interest. What you see here in the image is a trick that is very commonly used, and that is your eukaryotic expression vector also has somewhere in it sequence for a very peculiar protein called green fluorescent protein, or GFP. You may have heard of it before. As the name indicates, GFP is a protein that fluoresces green when you hit it with the right light, right? So not all cells in your culture will pick up the plasmid, only some will, but those that do pick up the plasmid will start expressing this green fluorescent protein. All you need now is a microscope that allows you to look at fluorescent cells under the microscope, and in a culture, you will see that some cells are expressing that GFP, some are not, like this cell neighbor to it. And now you have a very direct way of knowing this cell picked up the plasmid, and whatever I put in it, this cell didn't. And that can allow you directly to distinguish between these two subpopulations of cells, and then you can look at those cells that did incorporate the plasmid in more detail and start asking questions about morphology, shape, size, whether they're accumulating certain biochemical characteristics, whatever your question may be. As I said before, this is mostly in research contexts that is done, but it's a very direct way of distinguishing between transfected and non-transfected cells in culture, and then you can simply follow those that are expressing your marker and ask questions about them. Now, after 4, 5, 6 days a week, chances are that those plasmids are going to be highly diluted in your culture. So yeah, there may be 1 or 2 cells still expressing GFP, but chances are that the plasmid is gone. Not only because it became diluted, but also because in most cases it would have been destroyed by the transfected cell. Okay? Now what happens is this. And I need to take you back to something that we discussed when we talked about chromosomal rearrangements and aberrations. I don't know if you guys remember, but whenever there is 2 double-stranded DNA breaks in cells, which shouldn't happen, but it sometimes does, right? Cells are subject to all kinds of attacks all the time, and some of them may break DNA in 2 sites. What can happen is, instead of having non-homologous end joining such that you simply repair the 2 breaks and everything is as it was, what sometimes happens is end A here joins to end C, and the 2 ends of this B fragment join to one another, and you end up with a circular piece of DNA. At the time when we talked about this, we didn't really make much of those circular pieces of DNA. But now we understand that these circular pieces of DNA look a lot like plasmids. They look a lot like the vectors that we sometimes introduce through transfection into eukaryotic cells. Now, why does that matter? Because it turns out that whenever there is one of these circular pieces of DNA, what did I just tell you? That cells tend to attack them, tend to destroy them, because they think this is a foreign agent trying to infect me, trying to do harm. I'm going to destroy it. So most of those circular pieces of DNA, like the vectors that we introduce into eukaryotic cells, will be trashed. But every once in a blue moon, the cell goes into some sort of emergency mode that says, let's put this DNA back. It's a DNA repair mechanism that is basically saying, oh my God, there is a loose piece of DNA, we should put it back. And when the cell tries to do that, it can take 2 approaches. One is to go for random integration. It will put it just anywhere. The presumed evolutionary value of that approach, that desperate approach, is that there must have been some important information in that circular piece of DNA. Might as well put it back somewhere and keep it than lose it, right? So there are some cells that will just put it back somewhere in the genome, randomly. Other times, what the cells will do is see if there is any region of homology, any region of those circular DNAs that it can detect are homologous or share sequence similarity to other regions in their normal chromosomes. And if they find such homology, then there is a process called homologous recombination, by which the cell has a way to reincorporate that DNA into where it thinks it belongs. So it's almost like, oh, I recognize some sequence here in this circular DNA. I recognize similar sequences in one of my chromosomes. I'm going to put it there. I think it belongs there. That happens. It's an extremely rare phenomenon, but it happens. And as we would see in a few slides, it matters. So, every once in a while, cells, instead of destroying those vectors that we put in them, will actually incorporate them into their genome. And when that happens, which is going to be very rare, there may be 1 out of tens of thousands of cells that incorporated that vector into their genome, thinking it was DNA that always belonged to the cell and that it was recovering through these emergency mechanisms. It will incorporate it to the genome, And if you guys remember, these vectors also carry, I say if you guys remember, we didn't emphasize it much, but these vectors carry selection markers for eukaryotic cells. So the same way that you offer bacterial vectors ampicillin resistance or kanamycin resistance, some sort of antibiotic resistance, you do the same with eukaryotic vectors, such that later you can kill every cell in your culture except the ones that carry your selection marker of interest. So you can expose these cells to gentamicin, I think it's one of the most famous eukaryotic antibiotics, G418, any of those that will kill neomycin, that will kill all of the eukaryotic cells except the ones that picked up your vector, okay? And then it's just a waiting game. From one cell to tens of thousands, it may be several weeks, but if you wait the wait, it will happen. And now all of the cells have incorporated this resistance to the antibiotic into their genome and will remain resistant to the antibiotic forever. So you just, with patience and time, you basically amplify this single clone into an entire line that is now stably transfected. It has become what we call a stable clone. Okay? Questions, anyone? Yeah.
So it's usually going to start as transient and then something.
Correct. All eukaryotic transfections start as transient. If a transient transfection with that trick that I showed you before is enough for you, if you can, through microscopy, follow your transfectants and then ask questions that you can answer through microscopy, or if, let's say, you transfect your culture and you know about half of your cells are going to pick up the DNA, then you can have a non-transfected culture as a control, and whatever biochemical change, even if it happened in only half of the cells, may show up in your biochemical analysis, then you stop there. Your transient transfection is good enough for you. By the way, depending on the cell line that you're transfecting and depending on the transfection method that you use, labs have perfection techniques now that allow for upwards of 95% transfection rates. So this doesn't happen with just any eukaryotic cells, but there are some eukaryotic cell lines that allow you to transfect them with 90+% efficiency for only a few days, though. So you put in your genes of interest, you ask the question within a few days, and you get your answer. If, on the other hand, you're working with a difficult cell line, or you really need a pure population of cells, as we will see in a couple of slides, then you go through this stable transfection protocol.
Yeah. So have to keep retransfecting it until it...
Nope, because once the vector has been incorporated into the genome, that's it. Once it is part of the genome, the cells will replicate this vector as part of their DNA. So it just became one more of their genes.
Yeah, but to get that to happen, before that happens, you just
No. You do it only once, and the only thing is like you have to do with enough cells to start with, knowing that only in 1 million or 10 million cells will pick it up, but you play your numbers and you just wait. Yeah. So you don't have to keep transfecting, keep trying. Every once in a while, I suppose that it does happen, but generally speaking, if you have transient transfection rates that are up, close to 50% or so, and you start with 1 million cells, that means 500,000 of them are going to pick up the plasmid. All you need is one that incorporates it into the genome. Yeah. Cool. So now let's talk about how you go from cells in culture to a whole animal. Let's make a transgenic mouse. And what we will discuss about mice is applicable to other model organisms, except that because we've been working with mice forever in labs, it is generally with mice that people will do this, but you can do it with pretty much any mammal. So the process goes something like this. You take a pregnant female mouse and you extract a very early embryo from her really early on. And the very early mouse embryo will have this mass of cells in it called the inner cell mass. And that's a whole bunch of what we call embryonic stem cells. These are the cells that have the capacity to become anything in an adult mouse. Those are the cells that have the potency to be anything in the mouse, right? But what you do is you sort of dissociate those cells through enzymatic treatment. You plate them in culture, and now you have embryonic stem cells in culture. Okay? What you do then is take your vector of interest, meaning the one that carries your transgene of interest and some type of selection marker. In this case, I chose, because it's one of the most common ones, neomycin resistance. So neomycin is an antibiotic that attacks eukaryotic cells, not bacteria. Don't go around taking neomycin if you have a bacterial infection, because it won't be good. So you transfect these cells with a vector that has your transgene of interest and a selection marker. And it says there through electroporation, that's one of the tricks we use to put DNA into eukaryotic cells. What you do is you zap them with a shock of electricity, and that will create very brief pores in their cell membrane, and that's what the vector uses to go in. So you do that, and as we were discussing before, very infrequently, you hope that your vector carrying your transgene of interest and neomycin resistance will have flanking it on both sides sequence that is homologous to a specific sequence in the mouse genome that you would like to target for incorporation into the DNA. So what happens is, again, it's a numbers game, but what you're hoping is that your vector, I'm not drawing it here on the slide, but your vector is a circular piece of DNA that the cell will recognize as, oh my God, a piece of our DNA broke off. Maybe we should put it back. And what you're hoping is that when the cell recognizes those regions flanking your transgene of interest as sequences that it has somewhere in its own genome, it will incorporate it where the target sequence is through homologous recombination. Okay? So some cells in the culture will incorporate the transgene of interest into one of its chromosomes. And now what will happen is you have this mouse that is heterozygote for your transgene of interest, meaning one chromosome will have the transgene, we're calling it here TG plus for transgene positive, and then the other one, the other chromosome, the homologous chromosome, will have no transgene. Why? This process of homologous recombination is super rare, so it won't happen in both chromosomes. It will happen in only one of them, if you're lucky. Okay? The other thing that I'm mentioning here, because it matters to understand the process as a whole, is you start with a very special strain of mice that are black, that have like a black fur in them. We call them actually bulb C or black mice. And they have a black fur or really dark brown because they're homozygote for an allele that gives their fur that very dark brown pigmentation. So they're homozygote dominant B. Why that matters will become clear in a second. Cool. So now you have your culture of embryonic stem cells. Only one out of thousands of them has the transgene of interest as a heterozygote, one copy of it. Every other cell in your plate did not pick up the vector or picked up the vector and destroyed it. But that's fine. All you need is one, and then what you do is you subject that culture to selection with your resistance marker. In this case, we would add neomycin to this culture. All of the cells that didn't pick up the plasmid permanently, or the vector permanently, will die. And the one lucky cell that picked it up will survive neomycin and will keep dividing until you get hundreds or thousands of them. And all of them will continue to be heterozygote for your transgene of interest. Then what we do is we take those embryonic stem cells, and this is by far the hardest part, we inject them into an early embryo that we got from a different strain of mice. These mice are albino. So these mice are homozygote recessive for the fur pigmentation locus, so they're white, right? And they're homozygous recessive for the B locus, and what you're doing is you're injecting these transgenic cells into the early embryo. And I'm saying this is the most difficult part because it's very, very easy to damage and essentially kill these embryos during the process. So it takes a skill set that takes a while to develop. So you inject those cells into the early embryo, and the hope is that some of those cells that you introduced into these embryos will intermingle with the inner cell mass of that early embryo. So now you're going to have a mixture of embryonic stem cells, some of which are from a white background, and a few of which are from a black background and carry one copy of the transgene of interest. Did that make sense? Yeah? So within this early embryo, you have a mixture of stem cells, some of which carry the transgene and come from a black mouse, most of which do not carry the transgene and are from a white mouse. Good. When those early embryos, you re-implant these embryos into a female mouse, and when those embryos become pups, they're born, and what you get are these chimeric mice. You will notice that their fur has these patches of black or dark brown all around them, and that is because this mixture of stem cells will contribute to various organ systems in the mouse, such that when it comes to the skin, some of those cells came from the white background embryonic stem cells, and some of them came from the black background embryonic stem cells.
After injecting all that, does it become permanently stable, or can the mouse lose those traits over time?
It won't lose the trace over time because your DNA of interest has been stably incorporated into the genome of those very special embryonic stem cells that are transgenic, right? And once you have the chimeric mouse, some cells will have that gene in them, some won't. Now, what happens on the skin happens also at the level of the gonads. So just like you have a chimeric, patchy skin with some areas of white and some areas of black, if you could look, you would see that the gonads are chimeric as well. So the gonads, be the ovaries of these mice, if they're female mice, or the testes, if they're male mice, the gonads of these chimeric mice have two types of germline stem cells. These are the cells in the gonads that keep spewing out germ cells and eventually gametes, right? So some of them are going to be wild-type germline stem cells that come from a white background and have no transgene. And some cells are going to be transgenic germline stem cells, the ones that you got from the culture plate, if you will, from the culture dish. And those are going to be from a black background and will be heterozygote for your transgene of interest. Here's where I need everybody to take a deep breath because this is a conceptual leap. The idea here is that a chimeric mouse can produce three types of gametes. And it doesn't matter, it can be eggs if they're female mice, female chimeric mice, or sperm if they're male chimeric mice. There can be three types of gametes. There can be gametes that have a recessive allele for fur color and no transgene. That's all you can get from those cells because the ones that came from the white mice never had a transgene in them. There can be gametes that are that carry the dominant allele for fur color and no transgene. Those came from those gametes that did not inherit the transgene. They inherited the wild-type homologous chromosome. And then last but not least, there will be some gametes that have the dominant B locus and carry the transgene of interest. So that you know that these chimeric mice produce, you cross your fingers really, produce these three types of gametes. And the reason I say you cross your fingers is because the fact that you see patchy fur coloration doesn't necessarily mean that the gonad is patchy as well. You just assume that it may be. Right? So what you do is you cross these chimeric mice to regular white mice. These white mice, once again, don't have the transgene of interest, and they're homozygote recessive for skin color or fur color. This is almost like a test cross, right? Because what happens is if this mouse was producing some gametes with the dominant alleles in them, what do you think that progeny will look like? They will be black mice, right? Because if this mouse is producing gametes with only recessive alleles, and this chimeric mouse may produce some gametes with the dominant allele, if there were any progeny that is heterozygote for this locus, the dominant phenotype will show up, and those mice would be black. The reason I stress that is because what you do is you let these mice mate, they will produce progeny, and if you're lucky, and that chimeric mouse had a chimeric gonad and produced some gametes with the black allele in them, now you will have some black mice in the progeny. And these mice are going to be black because they're heterozygote for the B locus. Right? Now, among these black progeny or dark brown progeny, what you're going to have are some that do not carry the transgene, but some that do. Why? Remember that this chimeric mouse was producing, for every gamete that had the transgene, was producing also gametes that didn't have it, right? Because the transgene was heterozygote in this chimeric mouse. So, or in the original embryonic stem cells that gave rise to this chimeric mouse. So what that means is that, let's say we discard all the white mice because we know that those could not ever have the transgene in them. We keep only the black mice. Those black mice could come in two flavors, either heterozygote for the B locus and then wild type, no transgene, or they inherited a copy of the transgene of interest. In other words, these black mice could have two wild type copies of your target sequence in the mouse genome, or they could have one chromosome with a wild type copy of your target sequence and one that inherited that weird construct that includes your transgene of interest and neomycin resistance. How could you tell them apart? Excellent. Sometimes when I ask this question, I hear answers that have to do with give the mice neomycin and see which ones survive. It's not that easy. Your approach is much easier, less harmful to the mice, more ethical, right? So you're absolutely right. We designed PCR primers that would allow us to distinguish between a chromosome that carries a transgene and one that doesn't. One possibility is to design primers that target the transgene of interest. If you see a band, you're good. If you don't see a band, you're not. The risk with that is if you don't see a band, you're going to scratch your head thinking, well, maybe the PCR didn't work, or maybe there was something with the reagents, or maybe I messed up somewhere along the way. So a safer way is to design primers that sit just outside, such that, like when we were doing that fingerprinting for forensic science or paternity tests, if you get a short amplification product, it means there was no insertion. If you get a long amplification product, now you know that something inserted in between them. So that's what we do. We design primers that map just outside the target sequence. If there was no insert, the PCR amplicon will be shorter. That's what you see in this lane here. But if any of these black mice incorporated one of the transgenes, you should see that in addition to the short amplicon, there should be a longer one. So if this reminds you a little bit of those V1, V2, V2, V3, V4, V4 things that we discussed before, that's because that's exactly what it is. We are genotyping these mice. That's something that you may hear in lab environments a lot. Genotyping mice. What it means is, and the way we do it is, you mentioned take several samples. You don't have to, because these mice came now from gametes that carried this transgene. So if they have the transgene, it's everywhere in that mouse. So you don't have to take several samples. That you would have had to do in a chimeric mouse. But here, the whole mouse either has it or not. So the way we do it is we clip a tiny bit from the tail. It's totally harmless. They don't even notice. It's like clipping your nails. And there is enough DNA there to do these PCRs and find out if these mice carried the transgene or not. Okay? So, among these 4 hypothetical mice, which ones would you keep? Yell the numbers out. 2 and 4. If you're a little lucky, 2 and 4 are 1 male, 1 female, right? So now you have 2 heterozygote mice for your transgene of interest that you can cross to one another. And that's what you would do. You would take those 2, cross them to each other. If you're lucky, 1 is female, 1 male. And then you get this progeny here. And now you're going to see some white mice, but that doesn't really mean anything anymore because the white mice may come from combining the 2 recessive alleles. Remember that these black mice are heterozygote for the B locus. So these white mice may be homozygote recessive for the transgene, for the B locus, but still carry the transgene of interest. So ultimately what you want is you have this progeny and you go to town clipping tails and genotyping them, and what you're looking for is mice that now have an amplicon for your transgene of interest and no amplicon for the wild-type sequence. In other words, in a wild-type, you would have this short PCR amplification product. In a heterozygote, you will have both amplicons. In a homozygote for the transgene, you would have only the longer amplicon. Right? And these mice were heterozygotes, so what proportion of them are going to be homozygote for the transgene? Mendelian?
25%.
Louder?
25%.
25%, 1/4, right? This is essentially, when it comes to the transgene, a monohybrid cross. You're crossing 2 heterozygotes, you expect 25% of the progeny to be homozygote for the transgene. Here I say which ones are your homozygote transgenics, and like from here you know that it's any of those mice that have only the long amplification product.